All right. So finishing alleles, or at least moving on, I want to start with an interesting point that one of you made about the sickle cell anemia discussion board on Canvas. So this quote from one of you made a really interesting point, which I think is worth touching on briefly. There's a cost-benefit relationship with the sickle cell alleles. The wild-type allele gives you normal hemoglobin, and so your hemoglobin can carry oxygen to your tissues. What's the cost of being a wild-type plus over plus individual? What happens if you're a heterozygote? You're a carrier for the sickle cell mutation? What trait do you have if you're a carrier?
If you're just a carrier, you have protection against certain diseases.
Yep.
Malaria.
Malaria resistance. Okay. What's the third possible genotype? S over S. And that gives you a third trait, which is sickle cell disease. Yeah, sickle cell anemia, sickle cell disease. So there's wild type, there's malaria resistant, and there's sickle cell disease. Which one do you think is the best possible genotype of the three?
Heterozygous.
If you could pick, which one would you want?
Homozygous.
So you heard heterozygous and which homozygous? Okay, homozygous wild type. So why would those, does anybody vote that sickle cell disease would be the obvious choice for the genotype you'd want? Okay, good. So what's the difference between these two then, between plus over plus and plus over S? Why is one better than the other? Right. So the main problem with the heterozygous genotype is you have the possibility, you do have the possibility of passing on the disease-carrying trait to your kids. So there's a benefit and a cost, right? There's a cost-benefit relationship, as this student pointed out. So my question to you, just to think about, we're not going to talk about it right now, is if being a carrier is so great, if this is the best possible combination, or if that's the best possible combination, but if you're plus over plus, you and your kids don't get resistance to malaria. So depending on which one of these two, plus over plus or plus over S, you want to argue is the best, why aren't there more carriers? Why aren't there more individuals in the world that are plus over S, that are heterozygous for the mutation? So thought question. But in general, this was a fact that I think nobody said that this was not clearly a fact. Everybody found a paper that said that this fact, number 99 in the National Geographic article, was true. And there were loads of different publications that supported that, testing both in humans and also in mice and other animals to see what the effect of the sickle cell mutation was. All right, so today we're going to move into PCR, which is important for the study of alleles because this is one of the techniques that geneticists use to distinguish different types of alleles that different people have. It's a way that we genotype DNA samples. It's a really, really commonly used technique that if you do anything that's molecular biology based, not even just genetics, you'll probably be using PCR. Most of the biology department faculty members have these PCR machines in their labs because everyone's doing it in a good way. So most of these vocabulary terms, some of them we've covered already. Haplotype, genotype, homozygous, heterozygous. We've talked about homologs. We've talked about CODIS and locus. That was related to the genotyping, these tables of allele frequencies. So the parts that we haven't gotten to yet, denature, anneal, extend, primer, DNTP, PCR, agarose gel, electrophoresis, and the two at the bottom are all PCR-related terms. So that's where we're heading today. We haven't seen those terms yet, but we're going to get there now. And we haven't talked about non-disjunction, so I'm going to cross that off the vocabulary list for this topic. Anyway, we might get there eventually. There's also one of the exercises in this chapter that's specifically related to non-disjunction that we probably won't get to talk about. So far we've been working on the first task, and today we're going to get to the second and the third, today and next class. PCR primers, create a pair of PCR primers that will amplify a certain part of a chromosome. And we're going to finish up the last couple of exercises about the third topic, which is using these STR frequency tables to predict the likelihood of some individual suspect in a crime carrying a particular allele, set of alleles, haplotype rather. So that's where we're heading. Any questions before we get going? Okay. So one of the exercises for today was this question. What's the expected frequency of individuals that carry all three of those haplotypes? Size 10 at CSF1P0, repeat number 6 at TOPX, and repeat number 16 at VWA. How do you do that? How do you approach this question? We did an example of it last class. How frequent is the first haplotype?
0.232.
CSF1P0 is a region of a chromosome. Repeat number 10 has a frequency of 0.232. So about a quarter of individuals have that allele. About 0.25, about a quarter. Top X 6. Pretty rare, 0.004, about 0.4% of individuals have that allele. And VWA 16, where are we at? How many people in the population have VWA 16? 0.264. And these are importantly all on different chromosomes, which means they randomly enter sperm and egg in different combinations. So how frequent is a person that has all three of those, that combination? What's the mathematical process that we do? Independent frequencies multiply together. So whatever 0.232 times 0.004 times 0.264 is the answer. Is it a really big number or a really small number?
Really small.
It's a decimal times a decimal times a decimal, so it's going to be a pretty small decimal. What happens if I added a fourth haplotype to that list?
It would get even smaller.
So what's the implication for forensics? If you want to be really sure that your sample is guaranteed to match a possible perpetrator of a crime, if you're the prosecutor or the DNA forensic technician, what's the approach that they use to make really, really, really sure that the unique fingerprint is a match only to the suspect?
They use as many as they can.
Yeah, right. You genotype as many of these as possible because every one you add makes the probability that somebody is a perfect match that much smaller. Because every time you add another column to your calculation, you multiply it by another decimal. So the answer in this case is, let's see, 0.00025. So that's the frequency of that combination in. What's the caveat for this model? How is this not always 100% true? How does this sometimes not work?
In U.S. Hispanics.
Yeah.
It's in the
So
U.S. and there's just 50%.
this is the allele table for U.S. people of Hispanic descent. It'll look different if you look in the same paper. You have this PDF. It's linked from the course manual. You can go look at U.S. African American allele frequencies, U.S. Caucasian allele frequencies, and see how the numbers change when you look at the same STR, the same microsatellite, in people of different ethnicities. And we also talked about last time that this is just a sample from a representative. I still did not do what I promised I was going to do and look up if that n equals 140 means 140 people or 140 chromosomes. If it's 140 chromosomes, that would be how many people?
70.
70, 2 chromosomes per person. But it's a small number of people that are acting as representatives of the huge amount of diversity in the Hispanic population, much less the US African American population or the Caucasian population. Well, let's reverse this logic. What's the least likely haplotype combination of these two markers? Which one is the most rare? Which combination of TH1 and D5S818? How do you go about solving this problem? What's the rarest haplotype of TH01? So which whatever the smallest number is, which, yeah, looks like 10, 0.014. And what's the most rare D5S818? Allele 15, which is at 0.4% frequency. So if you multiply those two numbers together, which I didn't ask you to do, but if you did, that would give you the frequency of that combination of alleles. It's really rare. This will be on the next test. Hopefully it's not a shock. Something along the lines of using one of these tables to interpret, either predict most frequent, least frequent, do this calculation. Has anybody in here heard of PCR before? Few of you. Okay. That's short for polymerase chain reaction. The R, this is like ATM machine. You know about the ATM machines? They're redundant because M in ATM stands for machine. So we say it's like the Sierra Nevada mountains. The Mountain Nevada mountains. So people will often refer to the product of this approach. This is basically it's a solution we mix together in the laboratory, it does some DNA analysis, and the product is, we call it PCR reaction. It's the thing we work with. It's redundant. What is it? PCR is a technique that we use if we have a very rare DNA sample. Like you're in forensics, I know I keep coming back to that technique. There are other reasons you would use PCR as well. You collect a very rare sample that has very limited amount of DNA, but you want to do a lot of different types of tests on that DNA. You need to do something like take the maybe one copy of chromosome 2 that you collect from that one hair sample and make lots of copies, short fragments of DNA, from a particular part of that chromosome. Lots of copies, one region of one chromosome, one small region, usually 100 to 5,000 base pairs long, somewhere in that range. So relatively small, very small compared to the size of a typical chromosome. So why would we want to do this? Well, I already mentioned one approach is maybe that region of DNA contains, and we're going to work on this a little bit more, maybe that region of DNA contains one of these STRs. So this is how we get enough DNA out of the crime scene sample to ask, what is the length of that STR repeat? So we want to get a bunch of this DNA so that we can do something like DNA sequencing to determine the sequence of that part of the chromosome. And DNA sequencing can't work. We could spend a few classes on, but I won't at all. DNA sequencing doesn't work on one single molecule. You have to have lots and lots and lots and lots of molecules that you want to sequence to get enough data to know the DNA sequence of something. So that's one use of PCR. Make lots of copies of DNA for sequencing, for some sort of genetic analysis, and also for cloning. So people that want to take a gene out of one organism and copy it and put it into another organism also need lots of copies of that gene. And PCR is one way to get that. You isolate some DNA from a rabbit, clone the long ear gene, and then put it into a chicken. I don't know. Something like that. So it's a DNA technology that's good for, it's really common in molecular genetics, genotyping, and also in industry, making clones. Two things, this will hopefully be a review from the videos. There are two really critical, there are a lot more essential components. These are the most important two that we're going to start talking about today. Two things you have to do for PCR. And they were drawn on that, well, one of them was drawn on that previous screen, which is a piece of DNA that you want to make copies from. That's called the template. And where have we heard of primers before? Which was it? DNA replication. Because DNA polymerase needs a primer to start replicating DNA. So PCR is making extra copies of DNA, which means it's going to use an enzyme that makes DNA, which means it's going to use DNA polymerase. That is another essential component. Another bullet point, DNA polymerase, the enzyme that builds DNA. And it has to have primers to start synthesizing DNA. So in my previous color coding scheme, I'll just carry it on, red meaning RNA. So DNA polymerase needs the 3' end of an RNA molecule, or in this case, we're going to use DNA primers. It doesn't really matter. DNA polymerase can use either an RNA primer or a DNA primer to start replication. Our cells build RNA primers for polymerase to work with. When we set up PCR in the laboratory, we order these DNA primers from companies. The most famous one is in Coralville, Iowa, in case you care. I don't know, I guess they've got a big factory that just makes DNA primers. Single-stranded DNA molecules that everybody in the world that's a geneticist uses to set up PCR reactions. DNA polymerase can bind to the 3' end and then use a single-stranded DNA molecule as a template to build the newly synthesized second strand. So this process is DNA replication. We just do it in a tube instead of letting our cells do it. And we control which part of which chromosome we make lots of copies of by which primer sequence we use when we set up PCR. Because where the primers will base pair with a single-stranded piece of DNA, like drawn up here, is what dictates which part of the chromosome is going to get replicated. In our cells, primase makes primers at random in replication bubbles to prime replication. Here, we provide specific sequences of DNA so that we control which specific part of a chromosome we replicate. Any questions? Just getting started. Introducing the players. So let's see. Finally, I've asked this twice now. You may have recognized this question from a previous Socrative exercise. So PCR is similar to DNA replication. How? DNA is synthesized, B, absolutely. That seems to be the leader on the board. DNA polymerase is the enzyme that creates new DNA, also true. DNA polymerase in our cells synthesizes DNA. We take DNA polymerase, the enzyme, out of tubes in the laboratory to put in a PCR to replicate DNA. So C is also true. A is not. Why is A not true? In our cells, an enzyme does unwind our double helices.
Because in PCR, it really doesn't matter because there's no cell. The only enzymes that we need are the ones that we provide. And the DNA can, or the synthesis can start anywhere and there's no time constraint.
Okay. So we do provide, we, the scientists, when we do PCR, provide DNA polymerase to start this process. So what does the unwinding of double-stranded DNA in PCR? Heat. Good. Remember, there are two ways you can break double helices. You can either use helicase, the enzyme that unwinds DNA, or you can increase the temperature, which we've seen a lot, we've talked about quite a bit, to cause cause a double-stranded DNA molecule to become two single-stranded molecules. I goofed up. Usual. I think in the chapter it says central 10 base pairs, not 20, but it should have said 20. So this is a 30 nucleotide long piece of DNA. I'm asking, what DNA primer pair would you use to amplify the middle 20 base pairs, not the middle 10? So because I know this is 30 nucleotides long, I know the middle 20 is there, leaving 5 on either side. So there's the central 20 base pairs. So the question was, how do you design primers that flank this 20 base pairs to just amplify that segment, those 20 nucleotides? Say we're interested in those 20 nucleotides for some reason. It's an important gene that we want to clone. So on the next slide, what I've done is I've taken that original sequence and I've shown you its complement. This double-stranded piece of DNA. We're still interested in getting those 20 nucleotides in the middle. We want to make hundreds of copies of those. So that's the 20 base pair region. So if you were DNA polymerase, if you were trying to replicate the top strand, we've seen this before, it's just a new application. If you wanted to replicate the top strand, where would the 3' end of a primer need to be located? Anywhere up here. Where's it going to base pair with the top strand?
That T on the right side.
And why the T? So T on the right side, why there?
So then replication will start there and go in the direction.
So if you built this primer, ATGCG, so that it's complementary in sequence to the top strand at the 3' end of the top strand, what orientation is that primer? What's its polarity? From left to right.
3', 5'.
If it's base paired with the top strand, then it has anti-parallel orientation. So where's RNA polymerase going to be when it starts replicating?
At the 3' end of the bottom strand.
Right, so DNA polymerase will bind the 3' end of that primer, as we've seen before, and synthesize this direction. And when will it stop? Somebody say something soon. I'm about to run out of DNA. Quick. Oh, yeah, right there. So if that was the entire piece of DNA, if this, from 5' to 3', the top strand really was just 30 nucleotides long, that's where synthesis would stop. There's no more template, so DNA polymerase has nothing more to copy. So what we've done now is we've achieved a double helix based on the top strand as the template, and we've got a second strand that was just replicated. So now we have one double helix on the top. How do we do the same thing on the bottom strand? Where do we put the primer there that will synthesize a second strand of this molecule? We heated up this DNA. It fell apart into 2 single strands, the top strand and the second strand, the bottom strand, the 2 strands of the initial double helix. So where do we put the primer here? Where do we get a 3' end? Is it going to be on the left side or the right side of the bottom strand? So left side. What's the sequence going to be if we put some sort of primer here? Primer is going to has to base pair to that strand, so it has to have a complementary sequence. So what's the complementary sequence? From left to right, bottom strand, left side. Let's see, AGTCA. And that's anti-parallel polarity with a 3' there, which means DNA polymerase binds the 3' end and synthesizes a second strand from the bottom template, just like DNA replication. The only difference is we have bought the primers that we used, this 5' GCGTA on the top strand and the 5' AGTCA on the bottom strand. We bought those, we threw them into a solution with this DNA molecule, and we've added some DNA polymerase. We said, hey, DNA polymerase, go to work. make us a copy. So now we've gone from, in one cycle of PCR, we've gone from one double helix, the two initial strands, one double helix to two double helices. Right. The newly synthesized, this is now a double helix on top and a second double helix on the bottom. What happens next? We've gone from one copy of DNA to two. The chain reaction part, this is called polymerase chain reaction. What's a chain reaction?
That's repeated like 20 or 30 times.
One thing causes the next, causes the next, causes the next.
Snowballs, amplifies, like nuclear chain reaction, usually a bad thing, blows up. So the same thing is true of PCR. Well, it doesn't blow up, hopefully. If you're doing it right, it won't blow up. But it increases the number of molecules of DNA you have by doubling every time you do this process. We started with 2 strands of DNA, and after one round of synthesis, we've got 4. So now we have 4 strands of synthesis. So we repeat the process, add more primers, the primers base pair to their targets, and then we go from 4 strands to 8, and then 8 to 16 to 32, 64, 128, 256, 512, 1024, 2048. We do this 30 times, and you've got literally billions of molecules, billions of that short little piece of DNA in the tiny little tube. Billions. In like 3 hours. If you've never done this before, you'll see it if you get to Biology 104. That is, if you're biology majors, you will get to Biology 104. We do PCR in that lab class. And I know other lab classes do as well. So you will definitely be using this technique at some point during your career. So that's the chain reaction part, is that every time you add more primers and do this process again, you double the existing amount of DNA in the tube. 2, 4, 8, 16, 32, exponential growth. So with specific respect, though, to what we've been talking about, STRs and DNA fingerprinting, PCR is also used for that process. So take 3 minutes. Let me introduce this first. We'll go to the next slide, and then I'll give you 3 minutes to do something. So here we have, let's say this is your DNA. You've got 2 versions of chromosome 1 from your dad, from your mom. What's different about these 2 homologs? 2 different alleles, the paternal allele and the maternal allele. How are they different? Right. So there are parts that are similar. And of course, like usual, I've made this DNA sequence up for ease of demonstration. So there are parts of the chromosomes that are similar, and then there's an STR in the middle. There's a GC3 from dad, and an extra copy of that GC on the maternal chromosome. So the question is going to be, let's design primers that will amplify that middle part, just the STR, just the microsatellite, based on these flanking sequences. So design a primer, pair of primers, that you can use to amplify this STR from a heterozygote. This is a heterozygote, by the way, because it's got a different number. These 2 chromosomes have different numbers of repeats. 2 different alleles, GC3 allele, GC4 allele. So to make it easier, here's the template for the exercise. We've got the 2 different strands, the double helix from the paternal chromosome, and the 2 different strands, the bottom 2 from the maternal double helix. Take 3 minutes, design primers that will base pair with these single strands and copy across that STR. We'll come back in 3 minutes and we'll talk about what you've come up with. Do consult with friends if you like.